Hi, good morning, everyone. Can you hear my voice?
Yes.
Yes, okay, great. So, thank you all. We're gonna.
Good morning.
Hi.
Hi.
We're gonna record now this short session. Thanks for the people that are live. This will also be available as a short video as a part of the workshop that we're gonna do. I think we can just start. Good morning and welcome from BioDataScience101. This is the introduction to the third online workshop that we are doing as a webinar on the analysis of antibody sequences. So what we will do during the workshop will be to learn the data science and bioinformatics tools on how to analyze protein sequences in general. And in this short introduction, I want just to give a few information, a few indication on how to use the BioDataScience website and how which data set we are gonna analyze for this workshop. So let's quickly go. Let's quickly go to the BioDataScience website. Yeah. And I'll just log out and log in again to show how to do this. And you can see my screen, correct?
Yeah, sure.
Sure, great. So the BioDataScience 101 workshop use these cloud Jupyter Lab servers in order to make the experience very easy and straightforward for the students. So we have these cloud servers on that you can access using your browser, basically, and in that way you don't have to do anything. All the software that you need, all the packages, everything is already installed. The scripts are there, the notebooks are there, so you just have to log in and start working. You will be sent a username and password. I have my own, and then you can sign in just like this. And this is the main interface that you will be presented. At the beginning, no window will be open. Just a second, I will mute all the other people, the other participants. Okay. Super, great. So at the beginning, nothing will be open, but you will be presented with this interface, and you have on the left column browser, and you can see the files and notebooks and folders that are available. So this is just like an interface on your home folders on the remote server. All you need to do in order to get started is to find the modules. And for this class, we have two modules that I will present to you today. The main one, that it's this module, protein clustering. And there is also an introduction to Jupyter that we'll quickly go through. You double click on it and that opens. Now, I will not go into much detail with it, but if you're not familiar with Jupyter, Jupyter is a framework that allows to run Python and other languages like R and Julia in a very nice and interface that is mixing a markup, so comments and figures and all this kind of let's say metadata on the scripts that you have with the scripts themselves. The main interface is here in the middle, and you can just scroll it and go through it. Now everything is explained in the notebook, so I will not go into much detail, but you basically have two options. You can edit a cell. A cell, it's a small piece of information, or you can run a cell. And cells can be of 3 types. And the most common is the markdown, that it's for commenting. And you see here in this small menu, you see that this is a markdown cell, or it can be other type of cell like code, row. And to edit the cell, you just double click on it. And you see you have this kind of way to create formatted text, and it's explained in the notebook. So you can write whatever, delete it, and then to run the cells, you press Shift Enter. There are other ways to do it. Shift Enter is the quicker, or you just press the play button here. Now, up to this point, now it's not really that interesting. You can read it, but if you have a cell that contains some code and you run it, basically that will run on the remote server, and you will be displayed with the output. Now, this is very, let's say, some kind of very basic operation that we are doing, but you can do much, things that are more complicated. The notebook, when you change the notebook, it will be saved for you automatically, or you can just save from the menu or pressing Control S or Command S. And whenever you run code, this will be the interpreter. So it's called kernel that will run your code. So in this case, we are running code that it's in Python. Now, this is just a very quick overview. I will not spend more time with it, but I will go straight to the module on protein clustering. Just double clicking on it, a couple of seconds for opening. And this is the module that you will be working for this week. Now, this is probably, it's going to be, hopefully, that's going to be the last webinar that we're doing, because we are developing all these materials to have courses that are hopefully going to be in person. So people can sit down, working groups, and do all the activities all together. And the previous webinars that we have had were on reading and filtering data using pandas, that it's a Python library. They were on exploratory data analysis and on introduction to machine learning. This one, it's on a protein sequence analysis. This is a very broad field, and it's of a high interest to people that work in academia and people that work in industry. To give you an example, this is extremely relevant for people that now work on COVID. Basically, we are going to show tools that are state of the art for many of the tasks that we present. But this is also relevant for people that work, for example, in industry, because you have a family of enzymes and you want to discover the specificity of that enzyme, that it's a sequence analysis. You want to see which residues are conserved in a protein family in order to engineer that protein. That is something that you can do with the tools that we're going to present. And basically, I would say that a big part of bioinformatics is based on this kind of analysis, and learning how to do this in a reliable, robust, and automated way. It's really important for, it's a very important skill to have in your skill set. Great, so let's get started. I will now present the notebooks that we have, a quick overview of the notebooks. We are using an approach that it's called, that I call back to the basis. Especially for people that are already working, it's sometimes hard to get to acquire new skills, because probably, the way they are taught at the university is that, okay, you have a book or you have some reading materials and you have to start from scratch and reading a lot of things. And very often people don't have time for that. But they want also to be focused on the things that they really want to learn. So what we do here is this: we present a problem, and we start giving you the tools to solve the problem. Whenever you need a piece of theory or an extra reading material, that will be presented in the notebook. But what you will be presented is what is sufficient for you to understand the tool that you're using. Of course, sometimes you want to have a more, in-depth into the theory of that specific tool. Then we give you the opportunity with links, with other materials, to go through additional piece of information that you can use to broaden your understanding of the tools that we present. Because of these, the notebook is structured in a very specific way. As you can see, the notebook is also colored, and the color code is the following. In this kind of cyan blue color, you have the most important piece of information. So if you want to run through the notebook, the blue part is the part that you absolutely need to read. And then we also have questions that are used for you to get a feeling if you're on par with what you're supposed to know. And the standard questions are in green, and their advanced questions, if you want to get them more, challenged by the notebook, are in this kind of orange yellow color. Then we have the learning objectives, and as I told you, today we want to perform a bioinformatic analysis of a group of protein sequences, in this case antibodies, and I will present the data set in a few minutes. We want to be able to cluster the protein sequences in a meaningful way and to extract information from the cluster. And the tools that we are going to teach you are general tools for loading protein sequences into Python, filtering them, and cleaning the data set, and performing some basic alignment using a package that is called BioType. Then we are going to apply dimensionality reduction. There is a technique to visualize complex and multidimensional data in a simple way using 3 different tools. That's PCA, Principal Component Analysis, Disney, and UMAP. Then we will cluster the data that we have obtained using DBSCAN, that it's a very advanced clustering technique. And finally, we will visualize the clusters that we obtained and analyze them. In the case study that we're going to analyze, it's related to SARS-CoV-2. I will not spend time describing this. I think you've had enough of it, hearing about it. But we thought that that was interesting because it's actually a very good case study. And what we want to analyze today is the following question. We, in public databases, people have collected now thousands of antibodies that are specific either for SARS-CoV-2 virus or for closely related viruses such as SARS-CoV or other coronaviruses. And it would be interesting to understand whether the antibodies that bind specifically to SARS-CoV-2 are different from the antibodies that bind to other coronaviruses. And it's quite interesting because now, just yesterday, a paper was published that claims that, this is a paper from Italy that claims that in Italy, there were antibodies, so in the serum of people, that was collected much earlier than December or January 2020, when we believe that coronavirus SARS-CoV-2 first entered Italy. This paper claims that there are traces of these kind of antibodies much earlier in September, and they claim that in 10% of random samples they can find traces of such antibodies. But now the question is clearly, how can we be sure that those antibodies are really against SARS-CoV-2 and not against other related coronaviruses? Great. So, for example, in this case, we will see that there is a high overlapping in sequence between different antibodies from different, specific to different strains and different viruses. So probably the result that we are discussing is just that most of the population has already antibodies that are specific for other coronaviruses. It's still interesting because maybe people that have some kind of immunological memory towards other coronaviruses might have a milder disease then, because they already have some kind of pre-made defense to face coronavirus infections. And here there is a description and antibodies. I'm an antibody geek, so I could spend like hours talking about antibodies, but I will not do that, I promise. But what you need to know is that antibodies are the most variable proteins in our body. Basically, our genome only encodes for approximately 20,000 different genes, and the antibodies the repertoire at any given time in any person is approximately 10 to the 7 different antibodies. So their variability is huge. And this is the structure of an antibody. This is an IgG, that it's a classical type of antibody. And you can see it has this kind of Y-shaped. It has 2 light chains in blue, 2 heavy chains. And the variable part is this part here on the tips. And then the most variable part is actually here, the antigen binding site. That is extremely variable. So if we take random antibodies and we put them together, this region here would have basically very little sequence similarity between the antibodies that are not related. But when antibodies can bind to the same antigen, and the antigen is the molecular target of an antibody, these binding will produce some constraint on the sequence. For example, if the antigen, the target, is a positively charged molecule, the antibody will probably have negatively charged residues in order to bind strongly to that antigen. Of course, it's more complicated than this, but you got the idea. And the data that we are going to analyze, it's from the COPE-ABDAP database. That it's a database created in Oxford by the Oxford Protein Informatics Group, that it's led by Charlotte Dean. And we will have one of the group members in our webinar on the 25th to discuss about protein sequence analysis. From these, you have all your notebook. I will just quickly show you one thing, the kind of data that we have prepared for you. I just ran through the cells. Once you run, you see that this is the data that you have. And you see that you have this kind of table in which the information that we were going to analyze, the data that we're going to analyze are reported. And for each antibody, you have the name of the antibody, then antibody or nanobody. Most of the antibodies that we'll analyze are antibodies. Nanobodies are a specific type of antibodies that are only either present in camels or sharks, or engineered antibodies. And then the information that it's interesting, which virus it binds to, and which virus doesn't bind to. That is exactly the information that is important to understand the specificity. A few other information, then if it's neutralizing or not neutralizing, and then the specific epitope. And in this case, S means the spike protein, and RBD is a specific part of the spike protein that it's the receptor binding domain. That is the part of the spike protein that binds to the molecular target that viruses use to invade the health cells. And then they have the sequence of the heavy chain, sequence of the light chain, and a lot of other information. I will not describe everything, but the most important part here is what it binds to and the epitope. And starting from here, we will have a lot of other, tasks and things to do. I will leave that to you. I will now give you just a quick recap of what will happen. In the next few hours, we will send you a link so that you can log in to the server and work on the notebook by yourself using your pace. We are doing this because we saw that doing this in real time creates kind of a bit of a stress, because for some people, I mean, it might take 1 hour because they already are experienced with this kind of things. For other people, it takes 2, 3, 4 hours. They want to go more in depth. So you can go at the pace that you like. Then next week on the 25th, we will have again a Zoom meeting, but you will be asked to work in groups on a different notebook so that you can test the skills that you learned. And you will have a few hours until 12 to work on the notebook. And after that, we will have 2 short talks on how these kind of things are used in state-of-the-art research and state-of-the-art development of new drugs, for example. And then we will go through this notebook and have like an expert panel discussing the specific tasks and giving you some hints on how to do some things or what can go wrong when doing some kind of specific analysis. So I would say some kind of in-depth understanding of the tasks and the tools that you have been presented. That being said, I have finished my introduction. I just want to thank you for participating to this short webinar, and I'm looking forward to meeting you next week, 25th of November at 9 o'clock. So if there is any question, I will be happy to reply in the chat. And that being said, thank you, and have a great experience with BioDataScience 101. Thanks.